My name is Alexandra Krupski. I teach 9th grade biology at Staples High School in Westport, Connecticut. The course is really an introduction to biology. It's a little bit of everything. It's really up to the students to design the experiment and carry it out. Everyone's idea was a little different, which is what makes the lab so unique. They're really writing what I call an investigation article. The idea being that students, given the freedom and the opportunities, will learn how to write and develop really solid evidence-based arguments.
Why don't we label the cups then, like A, B, C, or 1, 2, 3, 4, and then test tube 1, 2, 3, 4, so they correspond with the paper cup?
Okay, sure.
We're really looking at how is carbon cycled in an environment. It's a safe enough lab where it gives them a lot of opportunity to kind of explore things and see how they work and how they don't.
So, cycling sheets out from yesterday.
Last class period we met and we did a pre-lab. They were put up into their lab groups and asked to read a very brief description of photosynthesis and cellular respiration and fill out a KWL chart. So what did they know coming into the pre-lab? What do they need to know to conduct the lab? And then they were given a way to research and figure out what they learned. They came into lab ready to go, having an idea of what an experiment would look like, the idea of the concepts and the methodologies behind it, and then it was their job to collaborate and actually carry out the experiment today.
Today we're actually going to be setting up and doing the experiments that we planned yesterday, and you guys did your homework.
So this was really an immersion, essentially, an immersion into inquiry and learning how to draft and write a scientific argument based on their experience today.
You got it.
They have their claim that they need to state, which is essentially a conclusion or a summary of their big idea, what was the lab really about, what were they testing, and then to give them planning time to meet in their groups and actually write out a procedure. So this is the information that I learned in my research, collaborate with other students in the group, and then go through and draft a rough procedure of what they would need to do to test their idea.
And you need to do the exhaling into a cup.
Oh, right.
Right, and then you can pour it into the test tube.
Blow through the straw into the solution for 60 seconds, we decided? Mm-hmm. Okay.
The next section that follows is their hard evidence. Their data tables, their charts, pictures from labs, some very brief descriptions of each thing. So it's really, how is your evidence valid, and can you support that or not?
So that means the oxygen didn't make it and photosynthesis is happening.
They had the second half of the lab period to really set up an experiment and collect data on their own terms.
So what are our observations? The liquid was bubbling.
Bubbling and started to change its tone and shade of color.
Okay, what happened with the plant?
The plant was, it started to kind of break off. Yeah.
Today was really their first fully collaborative experience. They all had to come together with stuff that they had researched beforehand, rather than just dealing with something that I've given them.
Test tube 1, no change. We should add that, right? There was no change
for any of them.
And then the longer piece is the reflection part, and that's really, does my evidence support my claim? Does my evidence count as good evidence? What would I do to make my evidence better? What did I miss in my data collection? What could I have changed to make the evidence stronger? I liked seeing them get excited about, having free reign of the materials and free reign of the lab and bounce ideas off each other.
Let's do the control first. Okay. So how many drops of BTB do we know?
Like 2.
There's no right or wrong answer. I'm really looking for how you defend your argument. How do you back up your analysis? What is the evidence that you are giving me to say this works or this doesn't?
Where's your chart? Oh, you could do that, sure.
Great.
That needs to get into your report, though, that diagram. But that's awesome. Nice job.
So taking away that fault, that right or wrong, opens up a safe place to take an academic risk in the classroom. I like watching them just explore. It's great.
But the control is the BTB with no plants, though. You are testing the plants, and you need something like the reinsurance of the bromomethyl blue itself, so...
I'm hoping that through this process, their writing becomes more developed. A lot of kids who come in and they're like, lab report is all they've ever written. They've never read a journal article. They don't see things like National Geographic or scientific publications as science. To have them come in with that mindset and to leave realizing that science is really about literacy and how you write and how you present yourself on paper is huge, because that's a skill that's going to carry them through all of the subjects for the rest of their lives.